environmental science? This is something related to
I
conserving water.
don't know, ma'am. Actually, I was all my teachers and my guided teacher has kept this in life science. I didn't know it was not supposed to be in life science.
Okay, okay. It's very good. If you must have been shown us like a working model, it will be very nice. It's just like a model which is not working. So what are that you have kept in tank, and below the next second tray, what you have kept? Any material which you have used?
Ma'am, this is the cardboard box, which is at
That
the
is model.
and
How it is, how the water is conserved?
ma'am, whenever we wash our hands, the water, whenever like after hand washing, the water is not completely used. It is still like clean water which we can reuse, but we don't reuse it. So many people don't find water and struggling to find water. So this idea can help them. Like the water from the sink goes into the storage tank, and then we can reuse the water again for flushing.
Okay. Okay, fine.
Thank you,
Yeah,
ma'am.
okay, fine.
Ma'am, actually, I asked to share screen my display sheets.
Yeah. Your display sheet is not available with the admin.
Oh, ma'am, but I've submitted it.
Yes, it's okay,
That-
fine, no problem. You understood your project.
Okay, ma'am. It
All
was pleasure meeting you, ma'am. Thank you, ma'am.
Question. One more question.
yes, yes.
Yes, sir.
Actually, how do you say that water used is clean?
Sir, when we use our hands, when we wash our hands, it can be cleaned. If any solid waste is there, we can I forgot to tell, we have a like filter which stops the solid waste to go in.
Which filter?
Solid means, sir, it is like straining it the water. So when
No, you are not clear. What makes the water clear? What is
sir,
special here?
sir, we can use chlorine tablets to make it cleaner.
What?
Chlorine tablets, sir, chlorine.
You didn't tell. Only when I asked the question, you are telling. Chlorine tablet will clean only the storage water, and not that the washed water. Then you have to. Are you treating it?
Sir?
Used water? Are you treating it with chlorine tablet?
Yes, sir. Inside there is a filter, right, sir? I just told you. So in that filter, chlorine tablet is present, and it also strains out the solid waste.
The chlorine tablet is not universal to clean everything, no.
Yes,
It
sir.
is only for specific for one particular microorganism. How come it can clean everything?
Sir, like chlorine tablet types of tablets, I was trying to tell.
You are not clear with that. Okay, you must be very clear what material using, and with that material, what type of impurities are can be filtered and cleared. You must be clear with that. Okay.
Sir,
Fine.
the germs which we use while washing our hands.
Okay. Fine.
Thank you, sir. And thank you, ma'am. We move on to our next candidate, MLS A6067, Sadiq Ali from MS Creative School, Akbarbad. Topic is measurement of glucose in our food. Sadiq Ali. You're there, Sadiq?
Good morning to one and all. Good evening, good afternoon to one and all. I am Mohammad Sadaqat Ali from class 7th of MS Creative School, Akbarbad Boys Branch. My topic is measurement of glucose in food. Why I have selected this topic? One day when I was having an ice cream, I asked with my grandmother, you should also have one, but she ignored and told me I am a diabetes patient. With this criteria in my mind, I have made this project to show the presence of glucose concentration in different food items and how it affects to insulin level in our body. Like most of the chemicals are present in our blood, the sugar level must be strictly controlled. This sugar level is controlled hormone in the pancreas. Sugar level, glucose level affect the sugar level in our body. This sugar level is controlled by insulin. My hypothesis is to check the different food items glucose concentration. And extract, we know that sugar makes sweet, but as we know that there are different kind of sugar, such as sucrose, the granulated sugar that we use usually for baking. Another kind of sugar which is found in the honey and in many different food items. Now we will see some of the glucose level in food items. First, we are taking a glucometric strip and a lemon. We are squeezing the lemon. See, the color is changing. See, the color has changes to the 250 mg per dl. So it contains low glucose level. We can use this in our diet. Now we will check this in potato. We will wait for some time. See, the color is changing. It has become dark. See, the color is changed. Till that time, we will, till the color change, we will see this in the rice. In this also, the color is changing. See, the potato one has changes to the darker one. The potato also contains the high glucose, which is harmful to our insulin level in our blood. Now we will see this in the ketchup. See, the color is changing. It is also becoming darker. With this, we have also seen in these food items also. Sugar can also contain the more glucose, and grape juice also contain more glucose. Honey also contain glucose, but it is a slight harmful for us. See, the color of the ketchup has changes to the darker one. Everyone says that the ketchup is made with the real tomatoes, but it is made with the sugar and glucose. With this, I conclude that my demonstration. Hope all of you understand how important is that to check the glucose level in our food. In this, I have made a logbook also. Jazakallahu khairan. Thank you. Hello. Ma'am, my voice is audible.
Yes, your voice is
Yes,
audible.
yes.
Judges, any questions?
No questions, ma'am. All the very best for you.
Yes, ma'am. Thank you, ma'am. Sir, any questions on your side? We go on to the next candidate.
Ma'am,
ML, yeah, yes.
ma'am, can I Display the devices I use for the project, ma'am. I'm Mohammed Fahim.
Your number?
Ma'am, I need to tell my number.
Yes, you have to tell your number, your code number.
MLS
Yes.
A
Okay.
007.
007. Yes. Judges, this was Mohammed Fahim from Unity Public School. He was not able to display his projects. I leave it to your discretion to decide whether we can give him the permission, ma'am.
Yes, same to display, madam. The hardness of water or the
Yes.
title.
Yes, ma'am. Yes, ma'am. Hardness, effect of hardness and acidity of water on plant growth. Yes,
let him
Fahim.
show.
Yes.
Ma'am, this is the device I use to measure the acidity level of irrigating water, ma'am.
Your video is not seen. Huh?
Ma'am, and my video is on, ma'am.
Yes, now it is
Can
on.
you see now, ma'am? So the white and black one is used to measure the hardness of the irrigating water, and the yellow and black one is used to measure the acidity in the irrigating water. So first, I would like to show the photo album, and this is how I use the pH meter. First, I calibrated using the buffer solutions and screwdriver given, and I tested it with the other, and after that I checked the results, ma'am. It was correct. And I checked for the hardness using this device, and
Okay, fine.
ma'am, can I show how to do?
We know, Fahim, it's okay.
Okay, ma'am. Ma'am, I would also like to show you my logbook.
Okay, Fahim.
Thank you, ma'am.
Can we continue with the next participant, ma'am?
Yes, ma'am. Thank you, judges. The next participant is MLSA 068, Simran Sultana from Government Girls High School, Golconda. Ziddi Dabon se Chutkara Pana. The topic is in Hindi. Simran Sultana.
अस्सलाम वालेकुम, मेरा नाम सिमरन सुल्ताना है। मैं जमात नहूम की तालिब इल्म हूँ। मैं गवर्नमेंट गर्ल्स हाई स्कूल सेकेंड क्लासेस में पढ़ती हूँ। मेरा टाइटल ये है कि कौन सा मुस्तही बेहतर है, यानी कौन सा डिटर्जेंट पाउडर बेहतर है, एरियल टाइड या सर्फ एक्सेल। मेरा मफरूजा ये है कि दाग धब्बों को मिटाने के लिए कौन सा डिटर्जेंट पाउडर बेहतर है, एरियल टाइड या सर्फ एक्सेल। हम रोजमर्रा जिंदगी में बहुत सारे डिटर्जेंट इस्तेमाल करते हैं, जिनमें कई डिटर्जेंट अच्छी सफाई करते हैं बनिस्बत उन डिटर्जेंट्स के जो अच्छी सफाई नहीं करते, दाग धब्बों को नहीं निकाल पाते। इसकी वजह ये है कि कुछ डिटर्जेंट्स में कीमियाई अशिया ज्यादा होती है और कुछ डिटर्जेंट्स में बहुत कम होती है, जिससे कपड़ों पर लगे हुए धब्बे अच्छी तरह साफ नहीं होते। इस मफरूजे को अंजाम देने के लिए मैंने ये तरीक़े अमल किया। कौन सा डिटर्जेंट बेहतर है, ये जांच करने के लिए हम पहले 9 रुमाल लेंगे। रुमाल को 3 हिस्सों में तकसीम करके पहले 3 रुमाल पर हम पैमाइशी तौर पर 3 चम्मच तेल लगाएंगे, कुछ इस तरह, तेल लगे हुए रुमाल। दूसरे 3 रुमाल को पैमाइशी तौर पर 4 चम्मच केचप लगाएंगे, कुछ इस तरह, केचप लगे हुए 3 रुमाल। और फिर हम तीसरे 3 रुमाल पर आधा कप चाय डालेंगे, कुछ इस तरह, चाय लगे हुए 3 रुमाल। फिर हम 3 किस्म के डिटर्जेंट पाउडर लेंगे। फिर हम 3 ग्रुप्स बनाएंगे। हर ग्रुप से कहेंगे कि वो 3 किस्म के डिटर्जेंट पाउडर से दाग धब्बे लगे हुए रुमाल को धोकर देखें। पहले ग्रुप से कहेंगे कि केचप लगे हुए 3 रुमाल को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखें कि दाग जाता है या नहीं। फिर हम दूसरे ग्रुप से कहेंगे कि तेल लगे हुए 3 रुमाल को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखें कि दाग जाता है या नहीं। फिर हम तीसरे 3 रुमाल को तीसरे ग्रुप से कहेंगे कि तेल चाय लगे हुए 3 रुमाल को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखिए कि दाग जाता है या नहीं। इससे हमें ये मालूम होगा कि कौन सा डिटर्जेंट पाउडर बेहतर है। फिर हम उनके बयानात को नोट कर लेंगे। ग्रुप ए ने कहा कि केचप लगे हुए 3 रुमाल को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखा तो एरियल से दाग बिल्कुल भी नहीं गए, टाइट से हल्के हल्के दाग रह गए, सर्फ एक्सेल से दाग अच्छी तरह साफ हो गए। ग्रुप बी ने ये कहा कि तेल लगे हुए 3 रुमालों को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखा तो एरियल से दाग अच्छी तरह साफ हो गए, टाइट से दाग बिल्कुल भी नहीं गए, सर्फ एक्सेल से हल्के हल्के दाग रह गए। ग्रुप सी ने कहा कि चाय लगे हुए 3 रुमाल को 3 किस्म के डिटर्जेंट पाउडर से धोकर देखा तो एरियल से दाग अच्छी तरह साफ हो गए, टाइट से हल्के हल्के दाग रह गए, सर्फ एक्सेल से दाग बिल्कुल भी नहीं गए। फिर हमने ये तजुर्बा किया, दाग लगा हुआ रुमाल, तेल लगा हुआ रुमाल, केचप लगा हुआ रुमाल। धोने के बाद चाय लगा हुआ रुमाल, एरियल से धोने पर दाग अच्छी तरह साफ हो गए। धोने के बाद तेल लगा हुआ रुमाल, सर्फ एक्सेल से धोकर देखा तो हल्के हल्के दाग रह गए। धोने के बाद केचप लगा हुआ रुमाल, दाग टाइट से धोने पर दाग बिल्कुल भी नहीं गए। इस मफरूजे से मैंने ये नतीजा किया कि एरियल बहुत अच्छी सफाई करता है, ये दूसरे डिटर्जेंट्स के मुकाबले में बहुत अच्छी सफाई करता है। मैं आप सबको ये सलाह दूंगी कि आप एरियल इस्तेमाल करें क्योंकि ये दाग धब्बों को अच्छी तरह साफ करता है। मैं ये नहीं कहूंगी कि आप सिर्फ एरियल ही इस्तेमाल करें, आप कौन सा भी डिटर्जेंट पाउडर इस्तेमाल कर सकते हैं, मगर मेरे मुशाहिदे से ये नतीजा किया कि एरियल बहुत अच्छी सफाई करता है। थैंक यू।
Ma'am, any questions?
Okay.
Ma'am?
What is her name? Yes. Her name
Her
is
name
what?
is Simran Sultana. Simran Sultana, ma'am.
We could not find her paper here or anything.
Yes, sister, you can ask in Urdu the question.
Yes. सिमरन आपका वीडियो ऑन कीजिए।
यस मैम।
आपने क्यों ये नुस्खा इस्तेमाल किया है, क्यों ये एक्सपेरिमेंट किया है आपने, एक्चुअली रीजन क्या है?
हम रोज देखते हैं कि हमारे छोटे भाई बहन बहुत सारे कपड़ों पर दाग धब्बे लगा कर आते हैं और बहुत जने बहुत सारे डिटर्जेंट इस्तेमाल करते हैं, मगर इसमें से कौन सा डिटर्जेंट अच्छी सफाई करता है ये जांचने के लिए मैंने ये मफरूजा लिया।
अच्छा, तो कोई इंडियन प्रोडक्ट या ऑर्गेनिक प्रोडक्ट इस्तेमाल करके देखना चाहिए ना कंपैरिजन के लिए। आप तीन मार्केट वाले प्रोडक्ट इस्तेमाल किया था, तो घरेलू एक प्रोडक्ट यूज करके आप कंपैरिजन देखते तो कुछ फायदा होता था ना।
क्योंकि लोग ज्यादातर यही इस्तेमाल करते तो इसलिए मैंने ये 3 लिए।
ओके,
मैंने
सो
ज्यादातर देखा
एक्सपेरिमेंट।
कि ये 3 ही इस्तेमाल करते लोग।
ओके, सो एक्चुअली प्रोजेक्ट जब आप बनाते हैं ना, आपका कुछ आइडिया से कुछ बताना चाहिए कि ये सभी लोग करते हैं लेकिन ये नुस्खा इस्तेमाल करना है। तो आपको खुद अपना रिसर्च या एक्सपेरिमेंट करना चाहिए। सुनाई दे रहा है? समझ
नो
में आ
मैम।
रही है बात?
नो मैम।
आपको सुनाई है? सिमरन आपको सुनाई दे रहा है?
यस मैम।
ओके, सो आप खुद कुछ बनाना चाहिए। ओके, मार्केट प्रोडक्ट से रिले नहीं करना चाहिए, आप कुछ बना के दोनों का कंपैरिजन करने पर कुछ फायदा होगा, वही प्रोजेक्ट होता है। ओके, सो इंशाल्लाह आगे कुछ ऐसा कीजिए कि ताकि आप बता सके कि घरेलू या ऑर्गेनिक और जो आप बना रहे हैं वो अच्छा है ज्यादा दर से, मार्केट वैल्यू से, मार्केट प्रोडक्ट से। ओके
ओके मैम, थैंक यू।
ओके,
Thank you so much, ma'am. The next candidate is MLSA 069 Rukaya Fatima from Garment Girls High School, Golconda. The topic is, does color affect the taste? Rukaya Fatima. You're there?
Government school.
Yes, sir. Government Girls High School, Golconda. Rukaya Fatima.
Good afternoon, ma'am and everyone. I'm Khaya Fatima from Government Girls High School, Second School. Today my topic is dust color effect test. What do you think dust color effect is? Let's see. My hypothesis is that color affects the taste when food is more concentrated. Let's start our activity. Here I have taken 3 glasses lemon juice, red, yellow, and green colors, marker and spoon. I use marker to write letters on the glasses A, B, and C respectively. Then I add the about 2.4 glasses of lemon juice in each glass. Line up all the labeled glasses. Add 2 drops of food color in the glasses and taste it. Then add more 4 to 5 drops of food color in the glasses and taste it. Now add more 8 to 10 drops of food color in the glasses and taste it. Note: while tasting drinks, cleanse the palate by drinking in some water and 50 ml. I observed that the solution gets concentrated. It tastes differently. My result is that after doing experiment, I found that food coloring affects the taste when it is used in more than its required time. My conclusion is that my hypothesis is practically matched. My result. Thank you.
Sollupa, sollu.
Rukaya, what are the things we have taken, ma? The tumblers? Is it the juice or only water you mixed it with?
Lemon juice, ma'am.
Lemon juice, then
Lemon juice, 3 different colors, glasses, marker, and spoon.
okay. So in lemon juice, you have added colors, food colors.
Yes, ma'am. Red, yellow, and green color.
Okay. So have you tested this with the volunteers?
Yes, ma'am.
Okay. So which one? What is the result?
I found that food coloring affects the taste when it is used in more than its required quantity.
Okay. The volunteers liked which color the most?
How many volunteers you selected? Okay.
Ma'am, please repeat the.
How many volunteers you have tested? With how many volunteers you have tested this? Ten, 15?
Myself, ma'am.
Okay. So which color was the best among these 3?
Three
Rukaya?
colors, ma'am.
So all the 3 colors were the best.
Yes, ma'am.
Okay, Rukaya.
Thank you, ma'am.
Ma'am, can we move to the next participant?
Yes, ma'am. Yes, ma'am. I now call upon the next candidate, MLSA 070, Maimoona Meveen from Alliance International School. The topic of project is treatment for ink stains by natural remedies. Maimoona Meveen
Yes, ma'am. I'm
from
here.
Alliance International School. Yes.
Yes, ma'am. So, good morning. Good evening. My name is Maimoona Mehveen from Alliance International School. There are so many dresses which we cannot wear because it is stained by ink. So, I got the solution for this. The title of my project is Removing Ink Stains by Natural Remedies. The problem is which mixture is best. I choose 3 mixtures: green papaya with vinegar, Colgate with baking soda, and bleaching powder. My hypothesis was green papaya with vinegar will remove the ink stains from the uniforms easily. So I did it like this. I took 3 pieces of cloth, white cloth, and I applied the ink on it, and then I applied mixtures on it, and I kept it like that overnight, and I washed it in the morning. And I washed it in the morning. The results were so different than I thought it would be. The bleaching powder worked more than anything. Green papaya was also worked, but it was 50-50. My independent variable is green papaya paste. Dependent variable is school uniform. But I suggest you guys not to use bleaching powder on colorful dresses because it will make a permanent white stain on your colorful dresses. You can use Colgate with baking soda instead on your colorful dresses. It will remove ink stains easily. Thank you, ma'am.
Okay.
Can you show the experiment there? Can you show the experiment there, the result? Water and how the stain removed or anything?
Sorry, sir.
The washed cloth, the stained cloth which you washed with the papaya paste and which you washed with the bleaching powder. Do you have that both the stained cloths with you?
Sorry, ma'am. I have it right now, but I have pictures of it. This was before and this was after.
Okay. So the treatment of the ink, that is the ink stain, was removed in the bleaching powder rather than the green leaf papaya paste.
Yes.
Why you have taken green leaf papaya paste? Green leaf papaya paste.
Because I want to use natural things. Actually, ma'am, I don't use green papaya leaves. I use the green papaya paste.
Green papaya paste. Okay,
Yes,
the
ma'am.
papaya fruit. You've taken the green papaya fruit.
Yes, ma'am.
It's not the leaves.
Yes, ma'am.
Why it triggered that only this papaya fruit is used for stain remover?
Actually, I want to use some raw fruits because the fruits which are ripe does not work. Like pomegranate, if we the pomegranate which is ripe just stained our dresses. So I want to use something raw fruits.
So papaya does not give the stain?
No, ma'am, it does not.
It does not give the stain.
Yes, ma'am.
Okay, Maimuna.
Thank you, ma'am.
Thank you, ma'am. We go on to our next candidate in the senior level, SLS A009, Nazreen Sulaika from Unity Public School with the topic Trigmotropism in tendrils. Trigmotropism in tendrils. Nazreen Sulaika.
Ma'am, is my voice
Nazreen,
audible?
you're there? Yes, your voice is audible.
1 minute, ma'am. Assalamu alaikum wa rahmatullahi wa barakatuhu. I am Nasreen from Unity Public School, here to represent about the topic stigmotropism in tendrils. So, we all know what is stigmotropism. It is nothing but to see how the plants show their response when any object is kept near the plant. So, when the plant senses the object, there will be a physical contact between the plant and the object, and this is due to the differential growth of the plant. This differential growth also helps in the stimulation of the plant in the particular region of the soil. So here, the non-contact cell starts to grow more faster, whereas the contact cells starts to compress, which results in the coiling response. So when the plant grows, simultaneously, the tendrils of the plant starts to coil around the object, which results in the faster growth of the plant. So due to this, auxins present in the plant, also, which helps the plant to grow more faster, is also being increased. So this stigmotropism, this process of stigmotropism helps in the good production of photosynthesis, where the plants are able to expose their leaves through sunlight and are able to grow more faster. So the reason why I took the topic is because nowadays the farmers face a lot of problems. So like they waste a lot of time in seeing while growing the plant or not. So with this process, they are able to grow the plants more faster. Jazakallah. Ma'am?
Yes, yes, yes. Nasreen?
Yes, ma'am.
Yes. So all the plants, you say that all the plants shows this stigmotropism?
Not all the plants, ma'am. At most of the plants, we can do the stigmotropism. Apparently. Like now I took the pea plant.
Only this family has the tendrils. The other plants do not have the tendrils. So you find only stigmotropism in the family Fabaceae alone. All the pulses family alone. What about the other plants? So you say that this will be helpful to the farmers. So the farmers which grow only these cereals or these pulses, for them it will be helpful, you're saying.
Yes, ma'am.
Okay, in what way it will be helpful, you're saying?
Can you repeat the question?
How it is helpful to the farmer by this stigmotropism? It is the inbuilt gene is there in these plants that they show the stigmotropism. In what way extraordinary it is going to help in the growth of the plant?
Like it can grow faster than before due to this process, ma'am. Like the auxin hormones will also be increased. So it gives a good production.
This process is surely there in these plants. See the cowpea which you have taken, the pulse, okay, pulse variety which you have taken, it belongs to the family Fabaceae, right?
Yes, ma'am.
So has this tendency and activity in it. It is nothing new in that. If you say this activity you can bring up in some other plant like mango plant or apple plant or guava plant, then we can say something. But this activity is there in this plant, cowpea plant. This stigmotropism is the gene which is present in the Fabaceae families. So surely it will have this sensitivity of whenever it comes with the contact. The tendrils, whenever they come with the contact or sense the contact, they'll start twining. You got my point, Nasreen, what I'm saying?
Yes, yes, ma'am.
Okay. Okay, Nasreen, anyway. Good. Your presentation.
Thank you, ma'am, for your valuable input. I call upon the next candidate, SLSA014, Khatija Nuha from Sana Model School. The topic is isolation of antibiotics from marine microflora. Khatija Nuha.
Am I audible and visible?
Yes, you're audible and visible.
Marina Beach is for making merry. How about making antibiotics? Assalamu alaikum wa rahmatullahi wa barakatuh. So this is me, Khatija Nuha, a 11th grader and a student of Sana Model School. So today, my topic is isolation of antibiotics from marine microflora. Before I get into the topic, let me tell you why should we do this? Why should I create antibiotics from Marina Beach? It's because excessive use of antibiotic has caused drug resistance. So to overcome this problem, it is necessary for us to discover new antibiotics. So this is why I'm creating a new antibiotic. So my hypothesis is to prove that marine microflora can be used as an antibiotic and it is sensitive to bacteria such as E. coli. So first, let me tell you what did I do. So on day one, I went to Marina Beach and collected soil samples from 3 different areas. So here are the samples. I collected the soil samples from 3 different areas. And I also collected the seawater. So after this, until they were treated, I stored it in the refrigerator at 3 degrees Celsius. So next day, I went to a food laboratory and started my project. So now I have these sediments here. So how do I extract antibiotics from these sediments? I need a medium, right? So I took nutrient agar medium. First, I prepared nutrient agar solution. Then I heated my soil samples at 80 degrees Celsius in aqua cleaning. Then I sterilized all the samples which I had, the Petri plates, the seawater, and these and the nutrient agar solution. So now it's time to make the agar plate. So I took 3 agar plates. Then I took the agar nutrient agar solution. I poured the solution into the 3 agar plates, and I left it for some time until it gets set. So after that, I took another 3 Petri plates and I took 1 gram of soil sediments here. Hope you can see. So I took 1 gram of soil sediments in 3 Petri plates. Then I diluted this with the seawater. I hope you can see the picture. So I diluted it with the seawater. After that, I poured these seawater solution into 3 mini bottles like this. I hope you can see in the PPT also. So I poured it into the solution. Then here I have the agar plates. So I poured my sediment solution, which I poured here, into the agar plates. So this was poured into the agar plates. After that, I kept this these plates into the incubator for 10 days. I incubated it for 10 days. Then I observed it on day 3, day 7, and day 10. So when I observed it on day 3, I wasn't able to see any growth on the agar plates. Then when I observed it on day 7, I was able to see good growth in all the 3 plates. So here are the pictures. So here are the pictures which I took on day 7. So I was able to see some tough powdery structure colonies on the agar plates. So this probably might be actinomycetes. I'm not 100% sure because I haven't done any further research. So it's my prediction that these colonies will be actinomycetes. So then when I observed it on day 10, the number of colonies gradually got reduced. So you can see the picture even in the PPT. The number of colonies on day 7 were more when compared to day 10. And when I compare the colonies in these 3 plates itself, I had good growth in sample 1 and sample 3. And sample 2 also had good colonies, but then it was less when compared to 1 and 3. So now I have the colonies with me. So how do I check whether these are sensitive or resistant to bacteria such as E. coli? Now we need to do antibiotic sensitive test. So with that, first I took the loop and heated it. I took the loop and I heated it and picked the colonies from the agar plates. I picked the colonies with the help of a loop from the agar plates. Then I suspended it into the saline solution. So here I have these samples. So this is the saline solution and I suspend the colonies into this. Then I got the sterile disc from the shops. Then I placed the disc inside the saline solution. After that, I made the Muller-Hinton agar plates. This time I didn't take nutrient agar, but I took Muller-Hinton agar plates. So I made the Muller-Hinton agar plates. Then I took a random bacteria from the food laboratory. Most probably that bacteria might be E. coli. So here I have the bacteria and here I have the agar plates. So with the help of a cotton swab, I took the microbial colonies and placed it in. I swabbed it into the agar plates. After that, I kept the agar plates into the incubator and I checked after 20 hours and after 28 hours. So when I checked it after 20 hours, I wasn't able to see clear zone of inhibition. It wasn't clear. I wasn't able to see proper zone of inhibition. So again, I waited for another 8 hours. That is after 28 hours when I checked the zone of inhibition, I was able to see good zone of inhibition. So this shows that it has the microflora which I isolated has moderate to mild amount of antibiotic properties. And in my project, my independent variables marine sediment samples. My dependent variable is growth of microflora. My control variable is sterile seawater, nutrient agar, Muller-Hinton agar, timing, and temperature. So with this, I conclude that my hypothesis to prove that marine microflora has can be used as an antibiotic is true. Yes, these microflora has mild to moderate antibiotic properties, and these can be used in aquaculture, and this will reduce the problem and overcome the problem of excessive use of antibiotics. So now my future enhancement. So in future, I'll do various biochemical testing and find it out whether it was a growth of actinomycetes or some other microflora. Then I would also isolate many pure cultures. And I would try the same project with different type of agar, like starch casein agar and different agars. So now it's time to thank everyone who helped me with this project. So first and foremost, this would not have been without the support of Allah subhanahu wa ta'ala. Then this project and the research paper and everything would not have been possible without the support of my teacher, Survat Jabin. She was there with me from day one, from the collection of the samples to this day. She helped me in everything and she cleared all my doubts. Then I would like to thank my uncles, my cousin Hania, my aunt, and everyone. They guided me, they helped me out in various ways. Then last but not the least, I would like to thank my friend Masiha, who was always there from the first day. So without all of them, I don't think so this would have been possible. Thank you. Any questions?
Thank you. A very nice presentation, Khadija. Ask a question or clarification. You should use the nutrient agar for isolating the marine microflora.
I'm not able to hear you properly.
Nutrient agar. So
Yeah.
not the nutrient agar is global marine agar is used for the marine isolation. So if you isolate the marine microflora, the global separate agar medium is needed for the isolation of marine microbes or microflora. Number one. Then next one, you should not confirm the actinomycetes.
Yeah, sir, I didn't confirm. I told it might be actinomycetes because I haven't done any testing. I mentioned it, sir.
Actually, some similarities are there. Actinomycetes similarities, some other floral populations are also there in the marine environment.
Yeah, that's what I didn't tell that it is actinomycetes. I just told it might be actinomycetes because of the powdery textures which appeared in the agar plate. So it was just my prediction. For that, I am not sure that it is actinomycetes. If I have to do further research on that.
And you isolate the marine microflora, you use only for the global marine media, not the a good result will be coming if you use the nutrient agar. Nutrient agar is used for the aquatic environment. Now, okay.
Yeah, sir, I use nutrient agar while.
Nutrient agar. Okay. Sure. Marine cultivating of marine environment, the marine microflora, global marine agar is used. Okay. We have a nice presentation. Okay. Ma'am, you are.
Yes. Khatija, you have done so much study, okay, then why you not analyze that what is the bacteria which is growing in your culture? You said actinomycetes. Actinomycetes is a big family.
No, sir. No, ma'am. I didn't tell it is actinomycetes. I just told it might be actinomycetes. I haven't done further research. In my project, I have just isolated the microflora, and then I have done the antibiotic sensitive test. Other things I will do in future, inshallah. Like the I'll find out which qualities were those.
Test. Okay. Antibiotic sensitive test you have done.
Yeah. Only this too was my main concept. This was in my hypothesis. Other than that, I didn't add anything in my hypothesis. I just wanted to find whether the marine microflora can be used as an antibiotic, and I wanted to test whether these are sensitive or resistant to bacteria. Only that was in my hypothesis. I didn't do any other research. That other things like whether which microflora was that and all I will do. That will be done in the future, inshallah.
Okay, Khatija, it was very nice. If you must have been mentioned at least 1 or 2 species which are present in that.
Yeah, ma'am. Yeah, but then I wasn't sure, right? I didn't do the biochemical testing, or I didn't do other testing. I only isolated it and I did the antibiotic sensitive test. I didn't do anything else. So that is why I didn't mention it. That will be my future enhancement, inshallah.
Okay, Khatija. Okay, fine. All the best.
Thank you, ma'am. I call upon the next candidate, SLS A015, Azari Jasmine from Fatima Central Senior Secondary School. The topic is Tenebrio mollitor, a detrivore. Azari Jasmine. Yes.
Hello everyone, my name is Azari Jasmine of class 12 from Fatima Central Senior Secondary School. My topic is Tenebrio mollitor, a retriever. My aim of this project is to analyze the efficiency of Tenebrio mollitor in decomposing different kinds of waste. I have taken this project because as the population increases, the amount of organic and inorganic waste also increases. To reduce this problem, I choose a retriever, Tenebrio mollitor, which can able to degrade both organic and inorganic wastes. For that, I took banana peel, boiler eggshell, and plastic cover as the different kinds of waste. Then my hypothesis is among the banana peel, plastic cover, and boiler eggshell, the Tenebrio mollitor fed with banana peel will digest earlier. Now my procedure is divided into two. One is diet preparation and another one is experimental design. In diet preparation, I prepare the diet by using the banana peel and plastic cover, boiler eggshell has the diet, and I took white bread as the diet controller, and I grind these mixtures into small powder particles. Then in experimental design, I took 4 containers, and I put these diet into different containers, and then I distributed 20 mealworms, 20 mealworms in different containers. Then I keep it for 1 week. Then after 1 week, I observe this. This is my result. This is my observation. In my result, I can observe that by keeping the bread as the mediator, I can easily conclude that among the banana peel, plastic cover, and boiler eggshell, the Tenebrio mollitor fed with banana peel will digest earlier. Then my conclusion is, though Tenebrio mollitor can degrade both organic and inorganic wastes, but more efficiently it can degrade organic has earlier. That's all, ma'am. Thank you.
Judges, any questions?
Fauzia, yes, your
Ma'am?
presentation was good. Yes, I don't have any questions, ma'am.
Thank you, ma'am.
Thank
Sir?
you, ma'am.
Nice presentation. Very good. Very good. No question, ma'am.
Thank you, sir. Call upon the next candidate, SLS A018, Al Jazeera Banu, also from Fatima Central Senior Secondary School, with the topic Best Natural Purifier, Moringa oleifera or Strinus potatorium. Hope I'm correct in my pronunciation. Jazeera Banu. Jazeera, you're there? Al Jazeera, you're there, ma'am.
Am I audible to you, ma'am?
Yes, you're audible.
Hello,
You're
everyone.
visible, you're audible.
Hello, everyone. I'm Al Jazeera of class 11th standard from Fatima Senior Secondary School. My topic is Best Natural Purifier, Moringa oleifera potatorium. In this project, my aim is to natural process in purification of water. Water pollution is a major pollution. It is a very harmful thing. When we use the polluted water, we will be affected. Living beings or drinks it will also be affected. When we purify this water, the pH, alkalinity, salinity will also be decreased. So we have to purify the water and we should use it. So my hypothesis is when we use Moringa oleifera, it will more efficiently purify the water than Strychnos potatorium. So my procedure is, first I took Moringa oleifera and Strychnos potatorium seed and I dried it for 3 days in the presence of sunlight, and I break the seed into small particles, and after that the 4 grams of Moringa oleifera I put it into 200 ml of distilled water, and after that the suspension was filtered with the help of a myelin cloth. Then for this study I have took 2 different types of wastewater. One is grey water and and other is fish tank wastewater. So the Moringa oleifera extract I put into those two different types of wastewater. After that, I mixed them vigorously and I made them to settle down. The same procedure is for Trichoderma sputatorum also. And after I have done this procedure, I found the turbidity, salinity, alkalinity, pH sulfate, and all these things. So you can clearly see that polluted and it is very much dirty. So after treating Moringa oleifera, it is purified very much. Likewise, when we use this in fish tank water also, the water will be purified. The same is for Trichoderma sputatorum also. When we use Trichoderma sputatorum in grey water and fish tank water, it will decrease the concentration of all these things. So, although these two things will purify the water, Moringa oleifera will efficiently purify the water than Trichoderma sputatorum. So, as a result from my observation, I found that Moringa oleifera will purify the water. Moringa oleifera will purify the water than Trichoderma sputatorum. So, as a conclusion, I conclude that my hypothesis is correct. So, as a natural coagulant, Moringa oleifera will purify the water more efficiently than Trichoderma sputatorum. That's all, ma'am. Thank you, ma'am.
Are you able to hear me, ma'am?
That's all, ma'am.
Yeah.
Fauzia Begum, do you have the sample which you have prepared?
No, ma'am. I have done this in a lab, ma'am. So only the observation I have done, ma'am. Here is the proof for that. You can clearly see that this is grey water, fish tank water. These two things were treated with Moringa oleifera, and this is fish tank water and grey water treated with Trichoderma sputatorum. This is the proof for that.
Why you have taken Trichoderma sputatorum?
So, Trichoderma sputatorum and Moringa oleifera seeds are mostly abundant in nature. We can see there these two seeds in street by street. So, it is more abundant in nature. So, it is low cost also. So, I took these two seeds, ma'am.
From where you got Trichoderma sputatorum seeds?
What, ma'am?
From where did you purchase these Trichoderma sputatorum seeds?
Trichoderma sputatorum seeds, I got it from my friend's house, ma'am. It was there in our house, ma'am.
How do you know that that plant name is Trichoderma?
It is a scientific name, ma'am. I already know it is its common name is Nirmali seeds, ma'am. So when I found that in Google and all, its scientific name is Trichoderma sputatorum, ma'am.
Why you have selected it actually? Because it is difficult to get this plant. It is not seen everywhere.
No, ma'am, it is seen in many villages, ma'am. If you see in many villages, it is most abundant, ma'am. I choose this Trichoderma sputatorum because it is also like Moringa oleifera. It will purify the water naturally.
That is, how do you know that? Did you see any reference or something that even this is helpful for the treatment of water?
What, ma'am?
Have you seen any reference that this also is used for the treatment of water?
Yes, ma'am.
Did you study any reference?
Yes, ma'am.
Okay. Okay, Jasiram.
One question, madam.
Yes.
Have you given the reference in the paper?
Yeah, in the link it is given, sir.
Have you mentioned the link in the paper in your?
Yes, sir.
With the title, have you given?
No, sir. It is in the column of topic of reference, sir. There, the link has been given.
See, you have given the many link.
What
link refers your point? There are many link you have given.
So I have searched for Trichoderma sputatorum and also Moringa oleifera, sir. Some things have been already done in these two, sir. Like Moringa oleifera, they have found that it is killing the bacteria like E. coli. It is already done. So I have referred those.
I'm not asking that. Whenever you give the link as a reference, you have to specify for what point, for what reference it is given. It should be numbered. Okay, one more question. how many times you treated and measured the water parameters?
So these parameters you are asking this pH salinity, sir.
Whatever you've done, no, you've given one data you've given. Okay.
Yeah.
It seems you've done once and you've measured, isn't it?
Yeah, sir. I have given after this putting the seeds, I have given this to lab, sir. So they have given the
Yes,
measured
that is what
values.
I'm asking. You've done only one sample, one treatment you've done, and one time you measured the parameter.
Yes, sir.
One time measurement is not reliable. You need to replicate, no? See,
No, sir. In the lab, they will be. They said that one or two times they have done this, sir.
No. That treatment has to be replicated. So you are treating the water and checking its parameter. The treatment itself has to be replicated. Okay, and thrice or 4 times. And then only, see, you are telling the pH 7.5. You have done once. One time measurement is not reliable. One time measurement is not reliable. Any experiment, any procedure has to be replicated to check whether it is by chance or
Okay,
it is
sir. In the
reproduced.
future, I'll do it for
No, it is not that. It is a basic. It is a scientific methodology. Whatever we do, no, we need to replicate and take the average value. Okay, fine.
Thank you, sir.
Thank you, sir. Thank you, ma'am, for your valuable inputs. I call upon the next candidate, SLSA 022, M. Nazreen Banu from Fatima Central Senior Secondary School with the topic antibacterial property of red dragon fruit on Streptococcus pneumoniae. Nazreen Banu.
Yes, ma'am. Assalamu alaikum wa rahmatullahi wa barakatuh. Myself, Nasrin Bonu of class 12, studying in Fatima Central Senior Secondary School. My topic is antibacterial property of red dragon fruit on Streptococcus pneumonia. We all love to eat fruit because it may produce vitamins and minerals to our body. But after that, what will happen? We throw the other parts like stem, leaf, peel. But it is also considered as bioactive components, which are helpful for our health. In this project, I aim to evaluate the antibacterial activity of different parts of red dragon fruit against Streptococcus pneumonia. Pneumonia is the bacteria to cause the pneumococcal disease. It can be spread while coughing or sneezing. It can also affect the body parts like lungs, ears, throat too. In this project, my hypothesis is peel, pulp, seed, leaf, the red dragon fruit, the highest antibacterial activity can be found in peel against Streptococcus pneumonia. In this project, my question is, what method should I use to detect the bacterial activity of different parts of red dragon fruit against Streptococcus pneumonia? Design of my study, independent variable, different parts of red dragon fruit. Dependent variable, antibacterial property, a control variable, Streptococcus pneumonia, red dragon fruit temperature. Material that I used for my project, nutrient broth, red dragon fruit, Streptococcus pneumonia, agar plates, cottons, distilled water, then lab facilities. This project done in the EHS 360 Lab Private Limited. First, I buy the red dragon fruit in a shop. Then I removed the parts like a pulp, peel, seed, leaf. Then it was dried for 1 week in a shaded region. Then dried part were ground in a mortar or pestle. Then the extract were obtained by maceration using the 96% ethanol. The antibacterial activity were carried out according to the conventional disk diffusion test using the cultures of Streptococcus pneumonia. The bacteria strain were cultured on agar nutrient medium. The brood media were inoculated for 24 hours. With the, for preparation of plate inoculation, 0.5 ml of culture were inoculated to 50 ml of agar media at 50 degrees Celsius. Then the agar poured into 120 ml Petri dishes, allowed to keep in the room temperature. After that, the microbial growth of inhibition was measured after 37 degrees Celsius in 24 hours using the instrument called digital calipers. These are my observation. The zone of inhibition of red dragon fruit parts for pulp 10 millimeter, for peel 16 millimeter, for seed 9 millimeter, for leaf 13 millimeter. These are the pictures during my after the zone of inhibition. This is my graph. From this graph, we can see that from peel is the highest antibacterial property, which has highest antibacterial property. My project shows the evidence to utilizing the fruit peel as source for natural antimicrobial, because it is the peel have the flavonoid. We can see in the last reference. I mentioned that in the last reference. From that reference only, I got that peel have the flavonoids. From flavonoids will do the will make the cells oxidative. It may reduce the causing of bacteria. I need to thank all my teachers who helped to produce this project to you. Thank you. Assalamu alaikum.
Okay, good one, good presentation.
Thank you, ma'am. Thank you, sir.
One thing,
Yes.
the peel as most highly active antibacterial.
Yes, sir.
Peel. Okay. Why you choose say dragon fruit for your study?
So because during my reference to which fruit can I select, in that I got the reference as red dragon fruit contains flavonoids. Flavonoid will make the bacteria to reduce the affection of disease pneumonia.
This fruit is not commonly available in our country. Mostly not commonly available. Anyway, you select the fruit. That's why I'm asking.
Yes, sir. Yes, sir. That's why I selected red dragon fruit. First, with this, we can have a pathway to other fruits like other jackfruit or mango, etc.
Okay, okay.
Thank you, sir.
Okay, Nasreen, very good presentation. Very nice project.
Thank you, ma'am.
So when you take next time, you try to compare and do it. Okay, so from the resource and reference, you identified it is the dragon fruit which has the peel has the more antibacterial capacity. Okay, so if you must have taken pineapple and compared with that, or jackfruit and compared, it must have been more useful.
Yes, ma'am. That's why in future I will also try this, ma'am.
Okay, ma'am.
Thank you, ma'am.
Okay, all.
I got idea to do the other projects. Thank you, ma'am.
Okay, ma'am.
Nazar sir? Sir, any questions from your side?
No.
Thank you, sir. The next candidate is SLS A024 S. Surega from Fatima Central Senior Secondary School with the topic Cultivo Bot. Surega. Surega, are you there? Yes? Surega?
Am I audible, ma'am?
You're audible and visible. Yeah, you can start.
I can't hear anything.
No, you're audible. We can hear your voice. You're audible.
Okay, ma'am, I'll start. So, good evening, everyone. I'm Surega from Fatima Central Senior Secondary School. I'm studying 12th standard, and my topic is Cultivo Bot. So, the main intention of this project is to reduce the workload of farmers and to reduce the usage and wastage of resources during agriculture. So, that's when we decided to make use of artificial intelligence. That's how we created this project. And for example, in the process of wastage of resources, I'll give you an example. That is, in agriculture, if there is a part of watering the plants. So if a farmer does the same work, he won't know the amount of water the plant needs. Sometimes it may be excess and sometimes it may be very less. But when a robot does the same thing, it provides exactly what the plant needs, and nothing is less, nothing is more. So like to save the wastage of resources, we came up with this project, and this can be particularly used in terrace farming. This is a model of the robot I made. I used Arduino to program to function it. This particular robot now can do plowing and sowing. I'll show you a demo. As you can see, this is how it works. We program the Arduino using C++ language, and also to conserve to manage the wastage of energy and other resources. We came up with the plan of making an organic fertilizer. That's what is there in my procedure. After preparing this robot, we tried to experiment it, and we compared it with the real life farming, plowing and sowing experience, and the result really amazed me. The thing is, the Cultivo Bot took 10 minutes to complete the work, whereas the real life farmer took 28 minutes to complete the same work. So I conclude it is also the time consumption is also very less. And also, the reason why I chose Arduino is that it's very cost efficient and flexible. So Arduino is a microcontroller, so whatever command I give it, that is how it functions. This same robot can be made to do watering and harvesting or any other work which I wanted to do. So this is how it works. If you have any question, you can ask.
Yes, you have said that you have done a demo, ma. In 50 square feet land you have done.
Yeah, ma'am.
Where you have done 50 square feet land, ma? In
our school campus, ma'am, terrace. We have a terrace farm where we have a terrace garden in our school.
Okay, so traditional method, how did you compare with the traditional method?
A traditional method is what we plow and sow, ma'am.
Okay.
It was done in our school ground.
So isn't it very small, which you have prepared the model? Cultivo Bot. It just took 10 minutes. Have you recorded correctly? When you take a 50 square feet land.
Ma'am, can you repeat, ma'am? I can't hear you.
See the robo which you have prepared, the Cultivo Bot, which you say. Okay.
Yeah, ma'am.
When you take a 50 square feet land, is it not small?
Obviously, it is small, ma'am. But terrace farm, this is particularly made for terrace farming and other stuff. So terrace farming is what we do in small scale of land. So I guess it's fine. The main intention of this this particular project, according to me personally, is I want youngsters to come forward and do farming, as it is very mandatory. We have to do it. Like, as I always say, farming is not a job, it's a way of life. So we have to practice it. So I thought this type of things may motivate youngsters to come forward and do that. So that's how we came out with this project.
Okay, Surega. All the best.
Thank you.
God bless. No question.
Aksasa, you have questions, Aksasa?
No.
Okay. Okay, ma'am. We'll move on to the next participant.
Thank you so much, ma'am. I call upon the last and final candidate for today's presentation, SLSA 026, Umi Habiba from MS Junior College, Malacca Pet. The topic is testing potential of hydrogen pH in beauty products. Umi Habiba.
Ma'am, shall I say?
Yes.
Good afternoon to one and all. I am Umm Habiba, studying in junior intermediate of commerce stream. I'm standing before you all to demonstrate my project, testing the pH of hydrogen in beauty products. So pH is the measure of how acidic or basic is the water. The range goes from 0 to 14, with 7 being the neutral. pH less than 7 is considered acidic, and the pH more than 7, greater than 7, indicates a base. pH is a measure of relative amount of free hydrogen and hydroxyl ions in the water. pH is reported in the logarithmic units. So the concept of pH was first introduced by the Danish chemist Søren Peder Lauritz Sørensen at the Carlsberg laboratory in the year 1909. Basically, this project is a research study to know the pH of grooming products that we all are using in our daily life. The question rises here is, why is it important that the pH of the product that we are using and the pH of our skin should match? So here I have done this project. I have tested a few very common face cleansers, hair cleansers, and soaps, which we all are using in our day-to-day life. The products I have used here are, first come the hair cleansers. I have tested nearly 20 to 30 hair cleansers. In the list are L'Oreal, Paris Repair 5, Dove, Clinic Plus, Garnier, Joy, Bella Indigo, Biotique, 9, etc. Their pH have come between 5.5 to 6. So they are good to use, but not that good how natural products do. And in the soaps, I have used here 11 tested over here, like Pears, Medimix, Dettol, and all, which are commonly found in our neighborhoods. So the good, not the good, the pH which is the least in this category is 5.5, which have come of Dove. So that is a good soap in this list. And face cleanser, I have tested nearly 9 face cleansers, and all of them are good. We can choose it considering our skin type. I can't suggest anyone because everyone's skin type is different, right? So I can say any face wash is good. It depends upon you and your skin type. And
the growth is either due to
the purpose of carrying out this research is to know the reality of beauty products, the brand themes, which they are satisfactory or not. And I am mentioning this that the pH values which I have got, I have done it at my home. They are not as correct as done in the laboratory. So I can't claim that these are the correct pH values. These are the assumed pH values by me. And the conclusion of this project is by testing this pH of the beauty products, I have got to know that what diseases can be trapped by using the harsh beauty products. Like triclosan is the harmful ingredient that is commonly found in the soaps, shampoos, toothpastes that we all are using in our day-to-day life, and it could lead us to the liver fibrosis and different type of cancers. So to safeguard us from all these sort of diseases, we should select the good and right brand which we are using for ourselves. And here I would like to acknowledge that this project is done by myself on my own. I'm not promoted by anyone or by my college. It is an independent project. And in this project, my mother and my brother have helped me a lot. Thank you so much. I'm done with my matter. Any questions?
Yes, Habiba. What are the products which you have tested for the pH?
Yes, ma'am.
Beauty products, you said. What are the products?
I have tested a few shampoos, face wash, and soaps, ma'am.
Your intention is to
I have made a
cover the
selection of it and tested with the pH strip.
nature of sand.
Where are those solutions in the pH strips?
I have taken some pictures of that, ma'am.
Okay. Okay, fine.
These are the results.
Okay, sir. We can.
Okay.
This is actually my photo album.
So all the creams you have used?
Creams,
or soaps?
yes, ma'am. All the things like which I use in my daily life and all the commonly found shampoos I have tested. At that time, I was not knowing to click the pictures, and afterwards that I got to know I have to keep the pictures also. I have clicked for a few products. Here is the list of all the products I have used. This is for shampoos. This is for face cleansers, and these are the list of soaps.
So what product more pH values you will get, and what products? Can you
Yes, sir.
product, whether it may be a facial creams or shampoo, the pH value elevated.
Sorry, sir.
So which product the pH value is more, is asking, whether in the shampoos or in the soaps or in
the soaps, ma'am, it has reached nearly, like you can see here in the soaps, it has reached near till 10, pH 10, which could really destroy our skin. And in the shampoos also, it has crossed the border till 8.9. But the most is in the soaps. Like here you can see the Santur Sandal soap. It has reached the pH of 10. So it comes in the category of soaps.
Okay. Okay, Abiba.
Okay, ma'am. Any more questions?
No, ma'am, no questions.
Okay, ma'am. Thank you very much. Have a nice day.
Thank you.
Thank you, judges. We are at the end of our presentation. I would like to extend my heartfelt thanks to Dr. Moideen sir and Dr. Mrs. Sheikh Azim for judging our candidates and sharing their valuable inputs. I extend my heartfelt gratitude for our judges. Thank you so much. We end our presentation session with this. Thank you, sir. Thank you, ma'am.
Thank you, ma'am.
Nia sister. Neha sister.
Yes, sir.
முடிச்சிடலாம்ல சார் சலீம் சார் சார்
End the meeting, sir. Sir, Fazal.
ஒரு நிமிஷம் சார், நான் இப்போ அந்த செஷன்ல இருந்தேன். ஓ ரைட் சார். முடிஞ்சு சார். Alhamdulillah, I would like to thank Dr. Sheikh Azim Taj and Dr. Moideen Askar Nawaz. Alhamdulillah, the interaction was very well, and you patiently heard from the student as well as raised questions. The session was very lively. Jazakallah khair, Dr. Sheikh Azim Taj and Dr. Moideen Askar.
Barakallahu feekum, sir. Thank you for the opportunity for us also, sir.
Thank you, sir. Thank you very much, sir, for giving me an opportunity.
Alhamdulillah. Fine. We can end the session. Huh?